students, welcome back to my channel. In this video, I am going to give you a brief lecture on oxidative phosphorylation. Oxidative phosphorylation is nothing but ATP synthesis is closely associated with the consumption of oxygen. So it involves the consumption of O2. So that's why this is called as oxidative phosphorylation. So it is based on Peter Mitchell's chemiosmotic hypothesis. So as I already explained in the previous video, so the translocation of electrons, the transfer of electrons from NADH or FADH2 to the molecular oxygen, so it establishes proton gradient across the membrane. The proton translocation occurs from the matrix to the intermembrane space. So that's why more number of protons, they are established in the intermembrane space. They are accumulated in the intermembrane space. Whereas less number of protons, they present in the matrix side. Okay. So this establishes a proton concentration difference. So then the protons, they tend to flow back to the matrix side. Okay, but the inner membrane of mitochondria, that is virtually impermeable to proton translocation. But it consists an enzyme complex. So we have already studied in the electron transport chain, that is ATP synthase, that facilitates the flow of protons into the matrix from intermembrane space to the matrix. Okay. So the F0 particle of ATP synthase that acts as proton channel. So that helps in the pumping of protons. When they are passing through the ATP synthase, for every 3 protons pumped through the proton channel, they release energy that will be utilized for the ATP synthesis. One inorganic phosphate is added to the ADP, then ATP synthesis occurs. So here you have to remember one thing, that is the protons are moving from the high concentration gradient to the lower concentration gradient. When they move down the gradient, so it releases some energy. That energy will be utilized by the ADP for the synthesis of ATP. In aerobic respiration, the NADH produced in the cytoplasm through the glycolysis, they do not enter directly into the matrix. So their electrons are transferred through the special shuttle systems. So there are 2 shuttle systems you need to know. So they are, first one is malate aspartate shuttle system. Second one is glycerol phosphate shuttle system. So let's see one by one. So let's see the first shuttle system, that is malate aspartate shuttle system. So NADH plus H plus are produced in the cytoplasm through glycolysis. They do not enter the mitochondrial matrix directly. They enter through a shuttle system present in the membrane. So let's see the shuttle system. So first shuttle system is malate aspartate shuttle system. Here the electrons from the NADH plus H plus, they are added to the oxaloacetic acid. Then oxaloacetic acid is converts into malate. So then this malate can enter freely into the membrane. It can be translocated into the matrix. So in the matrix, malate again converts into oxaloacetic acid. During this conversion, NADH plus H plus is regenerated. So these regenerated NADH plus H plus can enter into the electron transport chain directly through the quinone cycle. So directly through the coenzyme Q from the cytoplasmic NADH plus H plus, only 6 protons are generated in the intermembrane space. Okay. So let's see the second type of shuttle system. So that is a glycerol phosphate shuttle system. Here the NADH2 formed in the cytoplasm, they donates their electrons to the DHAP, dihydroxyacetone phosphate. So then it converts into glycerol 3 phosphate. Okay. So then it converts into glycerol 3 phosphate. It donates electrons to the FAD present in the inner membrane of mitochondria. So their FAD converts into FADH2. Okay, this FADH2 releases 2 electrons. They directly added to the coenzyme Q or ubiquinone. From there to they directly enters into the electron transport chain. Okay. So this type of shuttle system is present in the skeletal muscles and brain. Whereas malate aspartate shuttle system present in the heart, liver and kidney cells. Okay students, let's see the balance sheet of aerobic respiration. Okay students, let's see the balance sheet of aerobic respiration for one glucose molecule. Okay, so these are the steps involved in the aerobic respiration. So direct ATP produced in the aerobic respiration, NADH plus H plus produced in the aerobic respiration, FADH2, total ATP. Let's see how many ATP molecules are produced for one glucose molecule. Okay, so from glycolysis, so let's see the first step, glycolysis. So in this step, direct ATP are produced or 2 ATP are directly produced. Actually 4 ATPs are produced in the initial steps. 2 ATP are utilized, that's why so 2 ATPs in the glycolysis directly. So let's see NADH plus H plus production. So in the case of glycolysis, 2 oxidative reactions occurs, that's why 2 NADH plus H plus are produced in the glycolysis. So for every NADH plus H plus produced in the glycolysis, that means the location of glycolysis is cytoplasm. So they need to enter into the mitochondrial matrix for the ATP production. So they need to cross the inner membrane. That's why there are 2 shuttle systems are involved. Okay, so they directly enter into the electron transport chain, not by the complex one. They enter into the electron transport chain through the coenzyme Q, nothing but ubiquinone. Okay, that's why from every NADH plus H plus produced in the cytoplasm will produce only 2 ATPs. Okay, 2 into 2. 2 NADH can produce 2 ATPs. Okay, 2 into 2, that is equal to 4 ATPs. So there is no FADH production in the glycolysis. So let's see how many total ATPs we get from glycolysis. 6 ATPs. Okay, let's see the step 2. Oxidative decarboxylation of pyruvate. Oxidative decarboxylation of pyruvate, we didn't get any ATP directly, but we get 2 NADH plus H plus. Okay, so this occurs in the mitochondrial matrix. That's why for every one NADH plus H plus, we get 3 ATPs. Because 10 protons pumped into the intermembrane space. Okay, so 2 into 3 ATPs, that is equal to 6 ATPs. So there is no production of FADH2 in the oxidative decarboxylation. So total 6 ATPs are produced from the oxidative decarboxylation of pyruvate. So let's see how many ATPs are generated from the third step, nothing but Krebs cycle. So direct ATPs are generated, 2 direct ATPs are produced through the substrate level of phosphorylation in Krebs cycle, and 6 NADH plus H plus are produced. 6 oxidation reactions that producing 6 NADH plus H plus. So let's see 6 into 3. For every NADH, 3 ATPs are synthesized. That is equal to 18 ATPs are produced through the NADH plus H plus. So let's see the FADH2. 2 FADH2 are produced. For every FADH2, 2 ATPs are synthesized. So 2 into 2, that is equal to 4 ATPs. So let's see how many. 2 plus 18, that is equal to 20. 20 plus 4, that is equal to 24 ATPs are produced from the third step, that is Krebs cycle. Okay. So let's see the total number of ATPs are produced from one glucose molecule. Let's see, 6 plus 6 is equal to 12. 12 plus 34 is equal to 36 ATPs. So this is the net production of ATP from one glucose molecule. So this is the balance sheet, students. So let's see the energetics of aerobic respiration. So complete oxidation of one glucose molecule that yields 686 kilocalories energy or 2870 kilojoules energy. So the chemical energy released from one ATP in kilocalories is 7.6 kilocalories energy. So 7.6 into 36 ATPs, that is equal to 273 kilocalories energy. So the total energy is 686 kilocalories energy minus 273, that is equal to 412.4 kilocalories energy. This remaining energy would be released in the form of heat. So this is the energetics of aerobic respiration, students. So if you like this video, subscribe my channel. Thank you, students.